So hello, my name is Hirak. So I'm a course instructor in this metals in biology course. So this in this session, in this week 2 session, we I have framed a few questions for you guys, and we can just discuss a few questions and the probable answers. And we will basically this session is probably as you guys know that it's a problem solving session. So here I have framed a few questions based on this course, and I hope you guys like it. And so I am a PhD student under Professor Devruta Mighty at IIT Bombay, and I am also a PMRF fellow at IIT Bombay. So that's why I am a TA of this course. So if you guys has any query regarding any of the questions or anything, you guys can just mail me in this email ID that's mentioned in this first slide, and then so I will move forward with the few questions. So you guys can ask me regarding the questions, and you guys can type in the chat box, and we will move forward towards this session. I hope I am audible, right, to everyone. Okay, so the first question that I have framed is about which of the following enzyme requires ATP or adenosine triphosphate for its function. So it's like arginosuccinate lysase or arginase glutaminase or arginosuccinosynthesis. So this is basically about this arginine synthesis part, that how arginine is being about the enzymatic cycle of synthesis of arginine and its further derivatives. So if you guys know about it, then you guys can guess out which one could be a probable answer. So I would give you guys some time to figure out, and then I will show you the answer and we can discuss. So the probable cycle about adenosine triphosphate is arginase. So how do arginase require in this ATP? So the basic role of arginase is basically to form to break this urea-based bond to form orthonine, and basically it breaks this bond. So I will probably use a pointer here. Just a minute. So it basically breaks this bond, and they form urea in presence of water. So we get L-arginine, which is an amino acid, to a L-orthonine, and further we get urea. So in this whole, so the overall catalytic cycle of this process, which might look simple in this slide, but it's not that simple. So the overall catalytic cycle is like you have this arginine, which is a normally known amino acid. So here in presence of arginase, it produces urea and you get this orthonine. Now this further orthonine can be further degraded to citrulline, and further in presence of arginosuccinate synthesis, which requires ATP, is even giving you arginosuccinate, and further we are getting our arginosuccinate lysase, and that will further form fumarate. And so this is the whole catalytic cycle of arginase 1 and how it functions. So this enzyme basically requires your ATP to form, and further it will break because it requires energy, right? Because adenosine ADP is a high energy molecule. It has three phosphate molecules, so it the further breakdowns to give adenosine monophosphate uh, or its monomeric form. So the correct answer is B, arginase. So the second question is about liver alcohol dehydrogenase. So the main thing is which is the amino acid sequence that is being present in the active site of liver alcohol dehydrogenase. So you guys should know about liver alcohol dehydrogenase, that what is its function, and then regarding the metal ion present in the active site, and probably what are the amino acid residues that's present in the active site and playing some key role there. So I will give you guys some times and figure out, and then we can discuss. So the basic role, so if you see the liver alcohol dehydrogenase, so the enzyme that contains, uh, so the basic moiety in that molecule is, it contains zinc ion, and it contains various residues nearby it. But one of the very promising residues that is directly linked to the active site is this cysteine moiety with is two of the cysteine moieties. One is cysteine 46 and other is cysteine 174. And so therefore the only residue that really helps in the active site is this 174 and 46. So the correct option is answer A. And then there are also various residues nearby it, but those residues are not directly linked to the metal ion. So the overall mechanism of this liver alcohol dehydrogenase, as from the name it suggests, it's an alcohol dehydrogenase enzyme. So it's like So initially, if you have an alcohol, let's say you have this RCH2OH, maybe some long chain alcohol. So you have this zinc ion and you have this two cysteine moieties, and initially it presents this. The zinc ion is also bound with the water molecule. So that alcohol basically replaces the water molecule and it forms a zinc O bond. And further, what happens is there is a proton elimination from this whole molecule. And further, you got this zinc intermediate. And further, in the presence of NAD, NAD, NADH, and its further derivatives, it basically adds a hydride to the NAD plus and forms NADH. And so this is the only reagent that is being present in the is required in the mechanism to form from alcohol to aldehyde. And further, this aldehyde will basically leave from the molecule in presence of water to give your dehydrogenated product, or you can say our aldehyde. So the overall mechanism looks like this. And the moiety that is present is zinc 2 plus, and you have these two cysteine molecules directly linked to it. And there is also a histidine molecule, that is histidine 67, but the basic role of this cysteine moieties are very crucial. So we can move to the next question. I hope I am audible right to everyone. My voice is clear. Anyone can tell if my voice is not clear. Okay, so we can move, if it's clear, then we can move to the next question. So what is the sequence of histidine amino acid units in carbonic anhydrase? I have also discussed about this enzyme, I think in the first tutorial session. So if you guys have looked into it, so you guys can know about the amino acid residues present in carbonic anhydrase. Sorry, actually, as per the rules, I cannot speak in Hindi right now. So it's so you have to follow up the English. I think it's pretty simple and straightforward English. Um, you, I think you can see the slides, right? You can read the question. So if you see about carbonic anhydrase, so let's discuss about it. So if you see in carbonic anhydrase, the main residue, so the main role of carbonic anhydrase is basically to from carbon dioxide to your hydrogen carbonate. So that's the very basic role of carbonic anhydrase enzyme. So what it does is that whatever carbon dioxide we have from tissue, it takes, and in presence of water, carbonic anhydrase forms HCO3 minus, and further that can form your H2CO3, and basically that can further bind to hemoglobin. So the main moiety in the carbonic anhydrase enzyme that is present is zinc 2 plus, and it has three histidine residues in it. That's histidine 96, histidine 119, and histidine 94 that basically binds with zinc. And there are also other moieties present uh, on the other side of the zinc. So in the native state, it contains water in it, and further in presence of your carbon dioxide, what basically happens is this water uh, basically forms, and it basically eliminates proton and further forms a zinc OH, and that OH can further attack to the carbon dioxide to form a CO bond. And in presence of and there is a further conformation change in the zinc ion, so that can further leave in presence of water to your hydrogen carbonate. So the basic role of uh, this carbonic anhydrase is to form carbon dioxide to hydrogen carbonate. So the overall there are three histidine molecules present. That's your histidine 96, 119, and histidine 94. So the correct option is answer A. Okay, so let's move to the next question. So what is the polymorphic nature of porphyrin in hemoglobin? So I think you guys can know it. Yeah. But still, I will give you some time to figure out, and then we can discuss. So if you see the polymeric nature of hemoglobin, so I think most of you guys know that hemoglobin exists as a tetrameric form, as we have discussed in the previous class also. So it has two alpha chains that are similar, and it also has two beta chains which are similar. So hemoglobin is present in our blood, that's present in the red blood cells. So the overall structure of hemoglobin, as we have also previously discussed, it that it contains a porphyrin system, and there is an iron in the that is the active cell in the active center in Rn2, and there is also a free oxygen binding site from above the plane of the porphyrin, where the oxygen can basically bind, and we guys know that hemoglobin is oxygen binding enzyme. And further, there is also a histidine molecule that binds on the other side of the iron, and that is basically covalently bonded to the imidazole side chain. So hemoglobin basically exists in a tetrameric form. So that's the basic answer, and let's move to the next one. The number of aqua molecules and histidine units in dinuclear nickel hydrolase are. So what is nickel hydrolase, and how many aqueous molecules and histidine units are there? So this question is about the structure of nickel hydrolase. So if you guys can have studied about it, so you guys can figure out how many aqua molecules and histidine molecules are present. Yeah, so so let's discuss about the nickel dinuclear nickel hydrolysis. So if you guys know about nickel di then about the nickel hydrolysis, so it basically exists in various forms depending on the pH and depending uh, on the environment. But the very basic moiety that is basically present is this with which has two nickel, and there is a superoxo molecule, the oxygen, and there is also a carbonate molecule that basically binds with the two nickel. So the very starting, uh, the nickel exists in the overall complex binds with two histidine molecules with one nickel. So therefore in two nickel there will be four histidine molecules and there are two water molecules. Sorry, it should be water. And further, depending on the pH, it will form where it exists in various forms. So let's say here the two acetate molecule can go off depending, and it can form further bonds with water, and further it can also uh, get further reduced to various other forms depending on the pH and the environment. But the very basic state is this initial dimeric state, which has two acetate molecules which bind the nickel, and there is a nickel O and nickel bond. And first, so it has four histidine molecules and two water molecules. The next question is about vitamin B12. So if you know about the structure of vitamin B12 and what is the overall, what is the main moiety present and what is the metal, so you guys also should know about it, and whether vitamin B12 has a cobalt methyl bond or not. So that's the very basic question. So you guys can figure it out and then we can discuss. So, so let's yeah. So let's discuss about the structure of vitamin B12. So overall, if you see in vitamin B12, there is a cobalt moiety. It is present in cobalt plus one state. So it has this whole ligand that is present, and the cobalt is bound to a cyanide, that is CN. And further there is a side chain that further binds with your a sugar molecule, and further there is also a phosphate linkage between the whole system with the sugar molecule. So if you see the overall structure, there is no such bond of cobalt and methyl, but there is a bond of cobalt and cyanide. So the overall answer is that false, that there is no such bond of cobalt and methyl, but there is a bond of only of cobalt and CN, and there is also a cobalt bond with one of the other ligands that is present in the sugar moiety. So this question is only about the structure of vitamin B12. So let's move to the next question. So what is the active metal center in carboxypeptidase A? So if you guys know about the role of carboxypeptidase, what is the active metal center there, and what is and why is carboxypeptidase really doing? What is the role of it, function of it? Hey, can you please mute whoever is speaking? Please mute yourself. So if you guys know about carboxypeptidase, so carboxypeptidase also contains a zinc ion. So the basic role of carboxypeptidase is to break peptide bonds. So if you see about peptides, so basically peptides uh, are basically long chain polymers of amino acids. So carboxypeptidase basically breaks the C-terminal end of the peptide bond. So if you see this peptide bond has this C-terminal end. So here the carboxypeptidase A basically in presence of water, it breaks this bond and you get this polypeptide, which is basically N minus 1 because it breaks one of the amino acid, and you get this N minus 1 polypeptide. So in the active side, it contains zinc. It also and here are two histidine molecules, that histidine 69 and histidine 196, and there is also a glutamate molecule which basically helps in binding zinc at the active side. And obviously there is a water molecule that fills out the empty coordination side of the zinc ion. So the correct answer is zinc 2 plus. That is basically the active metal center in carboxypeptidase. So I hope let's move to the next question. So this question is about hemerythrin. So if you guys know about hemerythrin and hemocyanin, so these are basically oxygen transport protein. So you guys also should know about the structure and its function. How does it really function? So you guys just figure out whether it is an oxygen transport protein or storage protein, and so yeah. So I will just leave it for a time and for you guys to figure out, and then we will come back. So let's discuss about hemerythrin now. So hemerythrin is a basically oxygen transport protein. So hemerythrin is actually a non-heme oxygen transport protein. So if you guys know about hemoglobin and myoglobin, so that contains the heme molecule. But hemerythrin is or hemocyanin, which I will also discuss in the upcoming questions, is a non-heme iron oxygen binding protein that are basically present in mammals and bacteria. And the basic function of it is to is for oxygen storage and transport. So the overall structure of hemerythrin looks like this, which has two iron and there are two acetate molecules, and there is a OH linkage, and further there are histidine molecules that are binding with the iron. And further, when the oxygen binds, it basically forms this molecule, which has in iron 3 in both of the oxidized form. So the overall structure looks like this. So the binding of dioxygen to hemerythrin is basically here the oxygen binds with only one of the iron, not the other iron, but still the oxidation state of both the iron changes from iron 2 to iron 3. Okay, so let's move to the next question. So the next question is about hemocyanin. So you guys should also know about hemocyanin, how hemocyanin works and its function and its structure basically. So if you see about the structure of hemocyanin, so basically hemocyanin is also a oxygen transport protein. So it contains, instead of iron, it contains copper. And here also the same thing happens, like the copper oxidation state change from copper 1 to copper 2. But the thing that is different from hemoerythrin is that is the oxygen binding way of binding the oxygen. So here what happens is, instead of one oxygen getting bound to only one of the metals, here the oxygen bind in a bridge state, and here the oxygen binds both the copper, and the overall structure looks like this. So here you can see that hemocyanin are basically proteins that transport oxygen throughout some bodies in some invertebrate animals, and basically it contains two copper, and one more thing, it binds oxygen molecule reversibly. So here it can really basically also release oxygen if it is required at some stage. And one more thing, if you see about the oxidation state of copper, so therefore as the oxidation state of copper changes, it also will be the color. So when hemocyanin is basically blue when oxygenated and colorless when oxygen is removed. Why is it so? So the basic reason is probably the d electrons in copper. So if you see about copper 1, that will be d10. So therefore the all the orbitals are filled, so therefore there will be no dd transition, and so therefore there will be no absorption, so therefore there will be no color. But if you see about copper 2, that's d9, so there is a chance of dd transition that might be possible in the visible region, and that's why there will be a color. And basically in the oxygenated form, hemocyanin forms, it shows blue color. So hemocyanin is a oxygen transport protein. Okay, so let's move to the next question. The next question is, the storage of iron in human body is carried out by which enzyme? So whether it's myoglobin, ferritin, hemoglobin, or cytochrome P450. Yeah. Okay, so let's discuss about the solution. The correct answer is ferritin. So the storage of iron is basically done by ferritin, as we all know. But in human body also we have ferritin that basically stores the iron. So if you know about the structure of ferritin and all, I have discussed that in the previous uh, session. So the overall structure is like it contains iron in iron 2 plus state, and there are various amino acids in the active site. But the most prominent amino acids that are present is glutamate and histidine. So ferritin uh, is the enzyme that basically stores iron, and there is also one more enzyme known as transferrin, which basically transfers iron. Okay, so let's move to the next question. So next question is about myoglobin. So why is myoglobin used? Whether myoglobin is a catalyst for epoxidation reaction, whether it is a component in photosynthetic system, or it is a nitrogen fixation enzyme, or it is also a dioxygen binding metalloprotein. So what are the options that are correct? Okay, so let's discuss about it. So basically, hemoglobin is not a catalyst for epoxidation reaction. It is also not a component in photosynthetic system, and it is neither a nitrogen fixation enzyme, and we all know that nitrogenesis for that. So it is a dioxygen binding metal protein. It has the same system like hemoglobin, but it is basically a the monomeric uh, system. So it also contains porphyrin system where we have this iron and there is a oxygen binding in the benzene state, and there is also imidazole present from the other phase of the porphyrin. So it is a dioxygen binding metalloprotein. And that's it. Okay, so let's move to the next question. So this question is about how about the changes which occur when oxygen binds to hemerythrin. As I also previously showed you the structure of hemerythrin, so you guys can see what are the answers that are correct. Whether one iron atom is oxidized, both the iron atom are oxidized, or oxygen binds to one iron atom and is also hydrogen bonded, or oxygen binds to both the iron atom and is also hydrogen bonded. So what are the options that are correct among all these A, B, C, D? Okay, so the correct option is obviously you guys should know that both the iron atoms are obviously oxidized. That's option B. And there is also that the oxygen binds to one of the iron atom, not the both the iron atom, and there is also hydrogen bonded. So the correct option is answer A. That's B, and C is the correct answer. So as I previously also mentioned that both the iron atom is oxidized, and the oxygen only binds to one of the iron atom, and that is also hydrogen bonded. So you can see here it is hydrogen bonded with this OH bond. Okay, so let's move to the next question. So hemoglobin is an oxygen carrying protein. The correct statement about oxyhemoglobin is what? So it's about the spin state, it's about the oxidation state, how the oxidation state of the iron changes and how and whether there is a and also the space spin state of the metal when oxygen binds to it. Okay, so if you see about hemoglobin and the oxygenated and deoxygenated form, so it contains iron 2 in the deoxygenated form. So when oxygen binds to it, it basically forms iron 3 in the oxygenated form. And therefore it is also that the oxygen is in the O2 minus form because of this distal histidine molecule, and it is also in a bent form in the oxygenated form. So the metal ion, the oxidation state changes from 2 to 3. Dioxygen actually exists in O2 minus form or the superoxo form, and metal ion is in the low spin form, that's in plus 3 because of the that's in low spin, that's because of the weak field ligand, the oxygen ligands. So the correct option is option A. Okay, so let's move to the next question. The next question is about this porphyrin. So if you see iron porphyrin, it basically fails to exhibit reversible oxygen transport, and therefore it cannot differentiate between carbon monoxide from oxygen, as we all know that carbon monoxide is toxic. However, if you see hemoglobin, it doesn't have any of these pitfalls. So hemoglobin quite easily it recognizes the difference between carbon monoxide and oxygen, so it specifically binds only with the oxygen. But if you see generalized not hemoglobin, just the iron porphyrin, it cannot differentiate between carbon monoxide and oxygen, and also it cannot reversibly bind with the oxygen. So once it binds with oxygen, it that reaction is not reversible, so it cannot release oxygen at the later stage. So which among the following statements are correct regarding hemoglobin, why it can solve both the issues? So here the options is that iron 2 plus undergo oxodimer formation and the same is prevented in case of hemoglobin, or the second option that iron carbonyl bond strength is so much low in case of hemoglobin when compared to iron 2 plus state, or the third option is that iron CO is linear but iron oxygen is bent, and that is quite well recognized by hemoglobin. Or the last stage is that the interlinkages between the monomeric units in hemoglobin are responsible, which will basically overcome all these pitfalls that is present in iron porphyrin. So which among these correct, all these four options are correct, which basically helps hemoglobin to get out of both of these pitfalls? So if you see, the correct option is option B. So the first answer is that the iron porphyrin undergoes a oxodimer uh, formation, and that is prevented in case of hemoglobin. So why? So in case of hemoglobin, you cannot form a iron oxidimeric forming. So therefore, in free state, that is in iron porphyrin, what basically happens is that two porphyrin bridges to form a oxodimer formation. So if once the oxodimer is forming, so there is no um, free space to bind with oxygen. And the second option is that there is a bond strength difference between RnCO, and that is why they're in case of hemoglobin when compared to iron porphyrin. But I think that option is not correct. That's because the bond strength will be same in both hemoglobin and iron porphyrin, because that's just a iron CO bond. But the next option is that the iron CO is linear but the iron oxygen is bent. So that is quite well recognized by hemoglobin, and obviously that option is correct because CO is a linear molecule, so it basically binds in an iron CO so there is no such bond angle. But in iron oxo, as we previously also seen, there is a bond angle, and that is recognized by hemoglobin, and that's why in case of hemoglobin it specifically binds with only with oxygen, not with linear, not with CO. So that's the only reason why. So first option A and option C is the only reason why that hemoglobin can overcome both of these pitfalls. Okay, so let's move to the next question. So under physiological condition, oxygen is binding to deoxyhemoglobin and deoxymyoglobin. The binding curves and its pH dependence are what? So this question is about the oxygen binding curves of hemoglobin versus myoglobin. How is the curve, whether it's sigmoidal or hyperbolic, and also it's about the pH dependency of the curves, or the pH dependency of oxygen of hemoglobin and myoglobin, whether they are dependent or independent of the pH. So let's discuss about it. So under physiological condition, if you see about hemoglobin and myoglobin, so myoglobin is a monomeric protein enzyme, and hemoglobin is the tetrameric enzyme. So the very fundamental nature of myoglobin is that it can take only one oxygen molecule, but of hemoglobin it can take four oxygen molecules. But one more thing about hemoglobin is the cooperative binding. So if you see about hemoglobin, as more and more oxygen molecules keep binding, the more easier for it is to bind the next oxygen molecule. So that is why the nature of curve of hemoglobin is sigmoidal, but the nature of curve of myoglobin is hyperbolic. And one more thing about myoglobin is that it is pH independent. That because it can only bind one oxygen molecule, but if you see about hemoglobin, it basically depends on the pH as well. The and the as you can see here in this diagram, you can see here that this blue curve is for myoglobin and this purple curve is for hemoglobin. So the correct option is option A, that sigmoidal and pH dependent in case of hemoglobin and hyperbolic and pH independent in case of myoglobin. Okay, so let's move to the next question. So the next question is about nitrogenase. So if you guys know about nitrogenase that I have previously also discussed, so the reduction of nitrogen to ammonia is carried out by nitrogenase. So the question is about how many electrons are required in this process. If you guys know about nitrogenase, its structure and the overall reaction. So how many electrons are required in this process? So that's the very basic question. So if you see the overall process of nitrogenase, so the overall process is it requires 8 electrons. So if you see the overall process, so we all know that nitrogenase has a iron sulfur cluster, and it has iron and it has molybdenum in the structure. So the overall process is that it takes nitrogen and it fixes nitrogen to ammonia. So one molecule of nitrogen gives two molecules of ammonia, and it takes 8 proton and 8 electrons and 16 adenosine triphosphate or ATP molecule to produce two molecules of ammonia, 1 mole of hydrogen gas also. So the overall process is it takes one molecule and produces two molecules of ammonia, and it requires 8 electrons for it. The structure of this nitrogenase I have also previously discussed in the first problem solving session. And the overall process looks like this. It has iron sulfur cluster. It has a iron molybdenum moiety. So both of these moieties are channeling electrons to the nitrogen, and at the end it basically gives two molecules of ammonia. So you require, so the correct option is option 8, that it requires 8 electrons for this overall process. So let's move to the next question. Next question is about the changes which occur when oxygen binds to hemerythrin. So I previously also discussed that. Okay, so that's, I think, the repeat of the question. That it's both irons are oxidized and oxygen binds to one iron, and it is also hydrogen bonded. Okay, so let's move to the next question. The next question is about the ligand system present in vitamin B12. So I have also previously discussed about the vitamin B12 structure. So if you guys can see the main ligand system that is present in vitamin B12, just like if you see in hemoglobin, there is a porphyrin system. So in similar to that, what is the main ligand system that is present in vitamin B12, and what is its name? So the first option is porphyrin, or is it called choline, or it is called thala, or is it called phthalocyanine, or is it cronyism? So which among this is the correct answer? So the correct option among all these three, all these four, is option B, choline. So if you see about this choline molecule, so choline is quite different from porphyrin. So if you see about porphyrin, porphyrin has this methylene linkage or in all these four ends, which basically binds this four histidine type, or the five-membered imidazole sort of moiety. But in choline, there is one end where instead of this moiety, it directly binds these two moieties. So the overall structure of the choline is like that. And if you see the vitamin B12 structure, it is quite similar to that. It contains in one of the ends, it directly binds this five-membered histidine sort of moiety, and you get this direct linkage. So in vitamin B12, you have choline as the ligand system that is present in vitamin B12 structure, not porphyrin. Okay, so let's move to the next question. The next question is about the correct set of biologically essential elements that are essential to in our living organisms. So which are the elements that are very essential that you guys can figure or guess uh, that are very essential? The first option is iron, molybdenum, copper, zinc, or iron, copper, cobalt, ruthenium, or copper, magnesium, zinc, silver, or iron, ruthenium, zinc, magnesium. So let's discuss about it. So the correct option is option A. Iron molybdenum. We have previously also discussed that it is present in nitrogenase, and iron is quite present in a lot of moieties like hemoglobin, myoglobin, and copper is also present in a lot of other moieties like hemocyanin, and zinc is quite present in like carbonic anhydrase and liver alcohol dehydrogenase. But if you see about the other options, if you see like silver, magnesium, ruthenium, these are quite not biologically essential. So the correct option is option A. So the next question is about superoxide dismutase. As if you guys know about SOD, so it is quite abbreviated as SOD. So it contains which of the following metal ions? So superoxide dismutase is an enzyme. Uh, the overall structure of it is like copper and zinc. So superoxide dismutase is basically, as the name suggests, it is a major antioxidant. So if you see in our body, there are a lot of like UV rays. If you go out in sunlight, then the oxygen gets uh, to its electron reduced form, or the superoxide form, O2 minus form. So in our body there is a lot of enzymes like superoxide dismutase, whose main role is to act as an antioxidant or a defense system in our body against these highly energetic radicals. So the overall structure of superoxide dismutase is as shown here. It contains copper and zinc in plus 2 oxidation state, and it has the histidine molecules that binds with the in the active site. And there is also one histidine molecule that basically bridges along this copper and zinc. So the correct option is option B, that is copper 2 and zinc 2. So that's all for today. We have almost discussed like 20 questions so far. So I hope I have discussed and cleared all the questions according to your needs, and I think we can move. So if you guys has any question or anything, we can discuss, or we can end this session.